Bovine spongiform encephalopathy, BSE, or mad cow disease. Bovine spongiform encephalopathy, BSE, is a fatal neurodegenerative disease affecting cattle which belongs to a family of diseases termed the transmissible spongiform encephalopathies, TSEs, affecting a range of species such as scrapie in sheep and variant Creutzfeldt-Jacobs disease in humans. Jacobs disease, CJD, is a rare, degenerative, invariably fatal brain disorder. It affects about 1 person in every 1 million people per year worldwide. In the United States, there are about 200 cases per year. Testing for the presence of TSE disease in animals involves histological examination of hemotoxin and eosin, HE, stained paraffins, embedded tissue sections for evacuation of the neural pill in a method referred to as lesions, profilling in combination with assays to detect for the presence of PRPD. Hemotoxin and eosin stain produces colors different tissue structures, which would otherwise be transparent, so that you can get a detailed view of the tissue. As its name suggests, H&E stains makes use of combination of 2 dyes, hemotoxin and eosin. The 2 stains were independently introduced in 1865 and 1875, respectively by Boehmer and Fisher. The name bovine spongiform encephalopathy describes the characteristic sponge-like pathology observed in the brains of affected animals. BSE was first described as TSE in 1986 and reached epidemic proportions within the national UK herd during the 1980s and 1990s. Human variant Creutzfeldt-Jakob disease, VCJD, results from foodborne transmission of prions from slaughtered cattle with classical bovine spongiform encephalopathy, CBSE. Atypical forms of BSE, which remain mostly asymptomatic in aging cattle, were recently identified at slaughterhouses throughout Europe and North America, raising a question about human susceptibility to the new prion strains. Current mad cow diagnosis lies solely in the detection of late-appearing prions, an acronym for hypothesized geneless misfolded proteins, somehow claimed to cause the disease. In 1996, the first key epidemiological study on bovine spongiform encephalitis, BSE, appeared in the renowned journal Nature. In that article, it was estimated that by the year 1996, some 750,000 cows with BSE had entered the food chain in Great Britain. That same year, the first report on new form of Creutzfeldt-Jakob disease, variant VCJD, manifesting in young patients appeared. The number of new BSE infections in Great Britain peaked in 1991-1992 and has been steadily declining since. Characteristically, both spontaneous and familial CJD start in middle life, generally in patients over 50 years old. For decades, CJD was considered to be a non-infectious disease. Consequently, there were no precautionary measures to prevent possible infection of the physicians and contact persons. If samples appear to infect animals, it is impossible to prove that prions are causative. Lamiza's study showed that 55% of mice injected with cattle BSE and who became down with the disease had no detectable prions. Still, incredibly, prions are held as existing TSE dogma, and Hino Dringer, who did pioneer work on their nature, candidly predicts it will turn out that the prions concept is wrong. Many animals that die of spongiform TSEs never show evidence of misfolded proteins, and Dr. Frank Bastian of Tulane, an authority, thinks the disorder is caused by the bacterial DNA he found in this group of diseases. The neurotoxic, poison to nervous tissue as to the brain or spinal cord potential for cow tuberculosis was shown in pre-1960 England, where 1/4 of all tuberculosis meningitis victims suffered from Mycobacterium bovis infection. Tuberculosis is an infectious disease of humans and animals caused by the tuberculous bacillus and characterized by the formation of tubercules on the lungs and other tissues of the body, often developing long after the initial infection. Harley's study showed pathology identical to mad cow from systemic Mycobacterium bovis, a slow-growing aerobic bacterium and the causative agent of tuberculosis in cattle, causing a tuberculosis spongiform encephalitis. As a part of the funeral ritual, people in New Guinea ate brains of dead people. This resulted in a disease called Kuru, central nervous system disorder, which is associated with mad cow disease in cows. Villagers of the Fore people in the Eastern Highlands, predominantly women and children, would show the symptoms. In the 1960s, European scientists began to study people with the disease, called Kuru for the Fore word for shiver, and made two astonishing discoveries. First, that Kuru represented a new kind of infectious disease and that caused the brain and nervous system to degenerate. Second, that Kuru probably resulted from people eating their dead relatives. At the height of Kuru, 1 out of 50 people in some 4 villages succumbed to the untreatable fatal disease. In humans, the earliest symptoms include depression, insomnia, and anxiety. The person may look withdrawn and in a confused state of mind. Significant changes in the personality and behavior of the individual will show. The person may also experience muscle spasms, involuntary muscle contractions that are very painful. As the condition of the patient worsens, he loses muscle control and coordination, and may also face problems with vision and memory.